Good evening, everybody. I'm Vipin Vashishta. I'm one of the trustees of Child Health Foundation, and we are going to start this second edition of CHF COVID vaccine webinar. And as you know, India is in the midst of one of the largest vaccination campaigns in the world as far as COVID-19 is concerned. And already two vaccines, Covaxin and Covishield, have already been approved by the government of India for this purpose. And during first phase of inoculation, we are targeting healthcare workers around the country. There's a lot of confusion, apprehensions, and I can say that a lot of hesitancy amongst healthcare workers. It was least expected from this quarter, but this is the fact. And considering there are two different vaccines available at present, so there are a lot of discussions going on at various fora amongst doctors, also among the healthcare professionals itself, that which vaccine is superior and what are the side effects, when should we take it, can we defer it for a few months, similar many queries. And for this purpose, we have organized this webinar, and I am extremely pleased to announce that Dr. Jacob John, chairman of Child Health Foundation and very renowned virologist. I think for an Indian audience, he does not need any introduction. He has been the president of Indian Academy of Pediatrics as well as Indian Academy of Medical Microbiologists. He has spent several thousands papers in various national, international journals and various magazines, books. And he is one scientist who has very correctly predicted when the Indian leg of this pandemic will peak. And he is also now predicting that within 2-3 months, I think we shall be over and this pandemic will be over. And his immense interest as well as knowledge as far as this COVID-19 vaccine is concerned. And we have a very extremely eminent panelist for tonight discussion. And without wasting much time, I invite Dr. Jacob John here. Dr. Jacob John, sir, floor is yours now.
Thank you, Vipin. My audio is giving me a little bit of trouble, so you have to bear with me. We are planning this webinar in 4 sections. The first section will be Dr. Vasanta Muthuswamy talking to us on issues relating to ethics in the vaccine delivery program as currently going on in India. Then the second part will be Dr. Gagandeep Kang presenting the global overview of innovations in vaccine preparation, designing, and making available to countries like India. The third section would be Dr. Andrew Pollard talking to us about the Covishield vaccine and its mother vaccine, AstraZeneca. And we hope to have these 3 over and done with by 9:30. And it's because Andrew Pollard is busy, he's sort of allowed to come for half an hour with us. Dr. Gagandeep Kang and Vasanta Muthuswamy, as their time permits, they may kindly stay on, because that last section will be mainly for question answers. People would have many questions, and so if Vasanta, Gagandeep are available, please help me with fielding the question. Otherwise, Vipin, myself, Jagdish Chinappa, we will field the questions. So let me now introduce Dr. Vasanta Muthuswamy, who is an old friend for a long, long time. She retired from ICMR as the Senior Deputy Director General, and now she is President of the Forum for Ethics Review Committees in India. She is also the Founder Secretary and Steering Committee member of Forum for Ethics Review Committees in Asia Pacific and Chairperson of the Ethics Advisory Committee and Central Ethics Committee of Human Research in ICMR. She was responsible for various ethical guidelines of the ICMR, stem cells research, food safety evaluation, animal experimentation, etc. She has received several international awards, and she has written extensively in various books and many publications. So over to you, Dr. Vasundhara Swami.
Thank you. Thank you, Dr. Jacob John, for inviting me to this very interesting webinar. I know that everybody is very eager to know what's happening and what are the various options which are there at the moment. Of course, all of us know that the COVID-19 pandemic, it represents the greatest public health crisis since the influenza outbreak in 1980. But the speed and the volume of the clinical trials which have started in this area indicates that the need for production of high quality evidence-based therapeutic intervention. There are more than 300 clinical trials are going on, which are some are coordinated by WHO, but every country is having lots of clinical trials. I understand as per the WHO record, there are more than 300 clinical trials are going on and some are completed. But still, till date, there is not a single effective therapy which has been found for COVID. All those where the clinical trials have been done, most of them, the report says that they are not effective as such. And so nothing, no therapy is available. So the only option, and as of today, I think the latest record as of today, I just checked, it says that in India, we have total 1.06 crores of people who are positive, who are affected. And out of which till date, the deaths are 1,53,000 deaths have happened. So this is a kind of course, the number wise, it looks so huge. But of course, compared to the total population, if you take the percentage wise, it may be less. But still, we have more than 1 crore people who are infected and more than 1.5 lakh people who have died in this. And of course, the peak had reached in mid-September, and post-September, it has started coming down. So now we have a downward trend. Am I audible, Dr. John?
Yes, yes. No, yeah, I sorted it out. Yeah. My problem was solved.
Okay.
Yeah,
yeah, I think you couldn't hear, I think. Yeah. Dr. The others, I am audible for others, isn't it? Okay. So the peak had reached in September, and then of course there is a downward trend. But however, it is said that it was said that the only best way for all of us, for all the economic crisis and the health crisis which is happening, all those will be will come down and people will have come back to near normal if there is a herd immunity. Now this herd immunity, everybody is talking about it, everybody is writing about it. And if herd immunity has to really happen, unless I understand that 1 in 3 of the population gets infected, you can't reach a herd immunity, or at least 70% of people should be infected. So look at the number which has happened. I don't think herd immunity is possible in the near future. If you really think that, let the people get infected, and some of them, many of them will become sick, and some of them may die. And if you are going to wait for people to get infected in the community and then reach the herd immunity, it's going to take a very, very long time. It may not happen also. So the only other alternative seem to be, of course, internationally, everybody has agreed we have to come out with a vaccine very fast. So that vaccine is the best way of bringing in the herd immunity so that the people slowly can come back to the normal life. Now, if you look at of course, the international scenario, there are more than 50 vaccine trials are going on. And I think as of now, we can say that there are 6 vaccines which are available in the different countries, which all have been given emergency use authorization. And there are 6 which are available. Of course, all of you are reading about it. You all know about it. The first is the Pfizer vaccine, which is, and the second is the Moderna vaccine. Both are recombinant they are mRNA vaccines. Pfizer is supposed to be 90% effective, and Moderna is supposed to be 94.5% effective. Then, of course, we have the AstraZeneca vaccine, which is 70% effective. Then we are talking about the Sinopharma from China and the Sputnik from Russia. Both of them are approved in their country. Of course, they have all gone through phase 1, phase 2 only. Phase 3, as again, the results are not known. Both the countries have approved them straight away. And I don't think anybody has actually seen the data also of phase 1, phase 2 of these two, China and the Russian product. So, and we really, for most of them, even the Pfizer and Moderna, whatever efficacy data is available is very limited, done in a small number compared to what it should have been done. But of course, only those two vaccines, something is available. So now what we have in India are the two vaccines, the Covishield and the Covaxin. Of course, we have the another 3-4 vaccine trials have been approved, they are going on. But those which have been given now emergency use authorization has been Covishield and Covaxin. So if you look at both this vaccine, of course, Covishield has been straight away given the emergency authorization for implementation, and Covaxin, the DCJ has given an emergency authorization on a clinical trial mode. Of course, we'll just come to that little later. I just want to tell you, all these vaccines which have been approved in the different countries which are already implemented in countries, and the other countries where they have not, trials have not been also procuring these vaccines. Although we know that these are all supposed to be safe, immunogenic, and there is some kind of efficacy data is available for some of them, there are many things we still don't know about all these vaccines. We don't have a complete data for any of them. We don't know what were the original study endpoints for them, anybody to take a decision. And is it protective against both the mild type and the severe type? What are the long-term safety and efficacy issues? And what is actually the correlates of protection? How much should be the antibodies in the body to say that yes, these are going to be effective and protective? And how long the protection lasts? This we don't know about any of these vaccines. Whether one time vaccination is enough, or we have to take every year, or we have to keep on taking it every time when there is a infection happens, we just don't know. So these are the things which is not known about any of these vaccines. When WHO said that you can't consider all these vaccines going for phase 3 trial or for emergency use authorizations, they said there had to be 5 different characteristics which should be there. One is they say that the vaccine should have the ability to achieve herd immunity, in the sense they have to produce antibodies. And of course, it should be cost effective, it should be affordable for people. Then what is the ease of storage and the administration, how easy it is to administer? Whether, of course, they are talking about 1 dose versus 2 doses. And what are the safety profile, which is tests being done in different types of population. Similarly, efficacy also in the different types of population, young and the old and the adults and the vulnerable groups. Of course, we are supposed to know for all these things. And similarly, the efficacy also should be known. Now, so even if any vaccine is available, this can be only added to what is called the Corona toolbox, where the use of masks, the social distancing, disinfection, PPEs, we cannot do away with that at the moment. And in spite of having a vaccine, so vaccine, if it is given also, it is said that the Corona toolbox should be followed by everybody, it should be there. Now, when we come to these two, actually under this, if we really want to look at the emergency use authorization, and of course the WHO criteria for approving any vaccine, if you look at the WHO website and look at their criteria, it said that the safety profile, you can give the 25% percentage they have given for safety profile. Then the potential for efficacy, that means they should be able to produce antibodies, that is another 25%. And then they say the vaccine stability is 10%, vaccine implementation, the dose, route, and all those things should take care of 15%, and the vaccine availability should be 25%. So that vaccine availability, they mean by what is the ability of the different companies to do the scale of production, go for large scale phase 3, and then when the rollout is done, the vaccine will be available. I think these are the criteria which WHO has set up. Now, with all those things, and then when you really look at the emergency use authorization also, there are the stipulations are there. Since this is what is given in India, we have to look at what do they mean by the emergency use authorization internationally. Although that's a term which is not used in our NDCT rules. Of course, in the internationally, they are using that. I think the drug controller has also used the terminology emergency use authorization. In our guideline rules, it only says accelerated approval for drugs and any interventions. So if it's something has to be given emergency use authorization, as per the US FDA, they say that there are 3 criteria has been set up. First criteria is the product should be maybe effective to prevent COVID-19. They are not telling it should be effective. As per the US FDA guidelines, on the basis of which they have given approval to both Pfizer and Moderna vaccine, the first condition is the product may be effective to prevent COVID-19. The second is there are no available approved alternatives are available either for therapy or for prevention. And the third condition is the potential benefit of the product should outweigh the potential risk, either due to the product itself or due to the illness. So these are the 3 criteria on the basis of which emergency use authorization is given for all the vaccines, and this is there was applicable for everybody. So that's why we still, since in spite of the fact that we really don't know the complete data, the vaccines are getting approvals. Of course, in India, as I said, that we have not used the word emergency use in the rules. In the NDCT rules 2019, of course, much before the corona infection happened, they have introduced a class. This is in page number 186, 187. It's called the accelerated approval process after phase 2 if safety and efficacy is evident for an unmet medical needs. So if there is an unmet medical need for which there is no therapy or prevention is available, where after the phase 2 data is available about the safety and the immunogenicity or the safety and efficacy profile as per the phase 1, phase 2, approval, accelerated approval can be given for any product. And this is what is given in the 2000. I think this is what, I don't know because I'm not directly involved with the drug controllers committee, or so I don't know, but I presume this is what they are telling, that under this class it has been given. Now with this, when they are giving, let us look at both these vaccines now, Covishield and Covaxin, which have been given approval. Now Covishield, although it is AstraZeneca vaccine, but then AstraZeneca technology has been obtained by the Serum Institute of India, but they have produced their own vaccine. So it is an actually India-made vaccine, but the technology transfer has happened. They have obtained the technology from the from AstraZeneca, the Oxford vaccine, and it's produced in India. The trial which is going on, of course, they have done a phase 2, phase 1, phase 2, but it's in limited number. The phase 3 trial is still going on in about 16,000 people. And this trial is called Non-Inferiority Trial. So what they are telling is they are just still checking whether this Covishield produced in India with using the same technology as the AstraZeneca, whether this product is equal to the effectiveness of the AstraZeneca, the Oxford vaccine, we still don't know. So for that, the phase 3 trial is going on. They are terming this non-inferiority trial. So that information is also not available to my. So when I look at it, yes, I also feel that whether it is Covishield or Covaxin, for both we don't have phase 3 data. But the Covishield, I think approval has been given based on the international information which is available, where UK and Brazil, they have done the trial and there the efficacy data is available. Based on that, it's presumed since the same technology used, it should also be effective. Whether it will be effective, we'll know only after the phase 3 data is known. So when the emergency use rollout is given for Covishield, I feel that more or less both are in the same line, except that you know that the technology has shown some efficacy, but here, of course, we are waiting for the phase 3 data. We still don't know. So they must have looked at the, they say that on the international experience, they are giving approval for that. As I say, the Indian information is not available. When it comes to Covaxin, and of course, we have to see what are the different types of vaccine, like the Pfizer and the Moderna mRNA vaccine. The AstraZeneca is a chimpanzee influenza viral on the basis of which they have done that. And the Covaxin is an inactivated coronavirus itself, which has been taken up for the vaccine production. In the Covaxin, again, phase 1, phase 2 has been done. And the results are published in the BMJ Lancet. And that information was available to us to see. And I think they must have seen that also earlier. When it came to the Central Ethics Committee, of course, we had the chance to also look at the phase 1, phase 2 data, but it says that the dosage which has been taken for the phase 3 trial, according to that report, is 81.1% immunogenic. Immunogenicity has been produced in that, and that's what is the phase 3 trials are going on. So phase 3 trials are going on. Of course, we don't know the efficacy of it. So the issue here is, I was also equally concerned like everybody else, and how this approval has been given, and is it right without knowing the efficacy in a vaccine? Can we go for rolling out of a vaccine, are we doing the right thing when the phase 3, so another 2 more months when the vaccine, the efficacy data will be available, because all 26,000 volunteers have been given the first dose. I think maybe half of them haven't given the second dose. That means maybe another, by 2 months, it should be, the entire data should be available. So I was also, of course, worried about it. And then, of course, I also started thinking a little differently, thanks to Dr. Jacob John. Dr. Jacob John's interview in one paper had come, which of course I saw that. And then also he has written an article in the IJME. These two, when I read it, and I thought that, okay, let us also think differently as to why Dr. Jacob John, the most well-known virologist and the vaccine expert in this country, whom we all know very well, and he's so thorough about all those things. When he has made certain statement, let us see that what is it about it. And really, I started rethinking and looking at it. If you remember in your interview, Dr. Jacob John has said one thing, and he said that I am not worried about the efficacy of this, because effectiveness will come once vaccine is made use. I'm not worried about the efficacy, he said. So I just started thinking why he said he's not worried about efficacy. Then his paper in the Indian Journal of Medical Ethics was made available to all of us. When we looked at it, I picked up from your paper, Dr. Jacob John, 5 different points, which I think many of the other people also would have seen that. And I'll just tell you that what made us look differently in the situation when everybody was concerned and worried about what has happened, and what happened to the scientific expert committee which has given approval, and the drug controller has given approval. So in Dr. Jacob John's paper, I think many of you might have read it. In case people have not read it, I have picked up 5 important points in that. The first is that if protection, when huge number of people are affected and the infection is continuing, there is no treatment is available. If protection is delayed, then protection is denied. This is one strong sentence which he had given. The second sentence, the second issue is that if a vaccine is given after the epidemic is over, what use does it make? Is it really helpful? It has to be given when the epidemic is thriving. So it's good to do a fast track mechanism and revisit the rules for trials and make the vaccine available. So this was the second point, which was very interesting in what you had written. So I think probably, I don't know whether they've read it and they've taken it, and what it is. When I look at what has happened, when I look at your paper, I think these are the important points, that yes, we have to revisit the rules of approvals and then see that vaccine is made available as early as possible. The third point is immunological parameters is very important. And if the immunological parameters are detected during the phase 2, and if you know that the vaccine can be protective because it is able to produce the antibody, and that is a good surrogate for offering vaccines to the public. So I think that was one point. So these the vaccines seem to be producing, they are immunogenic and they are producing antibodies. Now, of course, another thing which you said, in case it has to be used, it should not be mixed up with the phase 3 trial. Phase 3 trial should go on because the data is necessary in efficacy. So when the rollout is done, it has to be done in a place which is away from the phase 3, so that the different people are affected, so that you can do a comparison and see that how it is working. And the last point, which is, of course, I picked up from this, is if it is found to be effective, if in the period, by the time the phase 3 data is known, if it is a matter of, say, 1.5 months or 2 months or a little more, at the end of it, if the vaccine is found to be effective, that means we have protected a large number of people by giving vaccination. If it is not effective, no serious damage can be done, because the adverse events are known, adverse events have to be monitored. And in case there are anything which has not happened earlier and it is happening, because as it happens when any product is goes into the community, that's the time when you know that the adverse events are always picked up, and that's why we do the post-marketing surveillance. Similarly, I think you have said that in case the additional adverse events are picked up, and of course we can always know that what happens when it is used in the large number. And of course it said that if there is reasonable evidence of protection and absence of harm, it can be morally justified. I mean, this is also you have written that. I think this paper I think has made everybody think a little, think about whatever is happening, whether it is right or wrong. I think so. When it came to the Central Ethics Committee, initially, of course, we were reluctant, and why should a Bharat Biotech product come to this committee? It's an ICMR Central Ethics Committee. For emergency use authorization in clinical trial.
Vasanta, sorry to interrupt you. Could you wind up pretty quickly?
Yeah, yes. Yeah, it is just over. So what was thought about it was that, yes, let us look at it. Since we know the safety profile, we know the immunogenicity profile, all of us have read those papers which are getting published. And there seems to be vaccines, both the vaccines seem to be safe. And in a clinical trial mode, we don't know what happens. So we decided what can be a clinical trial mode. So we should know that who are the people who are being given. There has to be informed consent has to be taken from them. There has to be monitoring of adverse event and serious adverse have to be done. Since the government is involved, government should be accountable for this. So it has to be a joint program by the government and the company and the ICMR, which is involved in this. So it has to be the accountability should lie with all the three agencies, and it should all the adverse events which are happening should be reported to the AEFI committee so that there is a national monitoring of all the adverse effects, whether it is Covishield or Covaxin. And in case a serious adverse event happens, since it is a clinical trial mode, the compensation is the responsibility of the company, with the company also accepted. So there is a 24/7 now helpline. It's not only under the AEFA and the COVID app they have given. The company has also given a toll-free number where anybody with adverse event can report. So this is what is our interpretation of the clinical trial mode. And we said, unless these are all taken care of, we are not ready to approve it. And immediately they agreed it will be done. And the way it is done, implemented now, I think all the suggestions have been accepted. So this is how, as far as the approvals and things are there, I think later on, when any question comes, I can give some more information if I have any. This is what, I mean, how we all looked into it. It's a committee of 15 members, and we had subject expert, Dr. Jacob John also we had invited during that person. And we had the subject expert. Everybody had a very long discussion, brainstorming session. And finally, it was said that, yes, let us do it. Very careful monitoring should be done. And then we'll know in the coming months what's happening. Thank you.
Thank you very much. That was a very comprehensive presentation of the tough decision-making process that the regulatory agencies and the ethics committee of the ICMR went through. Thank you very much. It's my pleasure to introduce Professor Gagandeep Pang, Professor at the Wellcome Trust Research Laboratory Division of Gastroenterological Sciences at Christian Medical College, Vellore. She served as Executive Director of the Translational Health Science and Technology Institute until 15th of August last year. For the last 2 decades, she has built a research program that has conducted key studies to understand enteric infection diseases in impoverished communities. She has widely published. She is known for the work on rotavirus vaccines, and she has provided immense boost to the science of vaccinology in India. Gagandeep.
Thank you, Dr. John, for inviting me to this meeting. I was asked to speak about CEPI and the global landscape. So I think Dr. Vasanta has given me information of which I was unaware about what the WHO and the FDA said about guidance for the vaccines. I was actually part of the WHO committee that developed the target product profile and the preferred product characteristics for the vaccines. But the weightage information that you provided was new to me. In terms of looking at what we expected out of a vaccine when WHO decided what needed to be done, it was really looking at it from the point of view of individual and population protection. And at the individual level, it was expected. We were not very ambitious. And we thought that we wanted a vaccine that would have over 50% efficacy. and it needed to be a vaccine that was safe, a vaccine that would provide protection for a minimum period of about a year, and that this would be a vaccine that could have, in the preferred product characteristics, we were a little more ambitious at 70%, wanting a vaccine that could be given to subgroups and that had reasonable storage conditions, could be considered as a single-use vaccine. As we've seen, the data from the Pfizer and the Moderna vaccines fulfill much beyond some of the characteristics that we expected of the target product profile. And when it comes to the FDA requirements as well, the FDA guidance that was provided as to the manufacturers, actually, I think it was pretty clear guidance that what needed to be done in terms of clinical efficacy studies, as well as the monitoring of immunological parameters. And in terms of the emergency use authorization that has been issued to Pfizer and to Moderna, it is based on efficacy data in a very large population, over 30,000 and 45,000 individuals that were enrolled in those trials. And they actually went well beyond the number of cases that they had anticipated for their entire trials. Since Andy has joined us here and he has a very limited amount of time, may I suggest that we have Andy's presentation? I have plenty of time this evening and I'd be happy to talk later. Dr. John, over to you. Would you like Andy to speak now and I can pick it up later?
Thank you, Cherry. Welcome to Andrew Pollard. Good evening. Cherry, could you tell us a little bit about CEPI?
Sure. CEPI is the Coalition for Epidemic Preparedness Innovations, and CEPI was established after the Ebola pandemic. In Ebola, the actual trials of the vaccine started when the epidemic was waning, because it took a long time for people to organize themselves and plan the trials, get going with the trials. And this despite the fact that the vaccine had actually been developed almost 15 years prior to the Ebola outbreak in West Africa in 2014, and that technology had been transferred to an American company. So in the aftermath of Ebola, there were a lot of global consultations, there were a lot of editorials written, and a grouping that consisted of the government of Norway, the Gates Foundation, the Wellcome Trust, the World Economic Forum, and the Department of Biotechnology came together to be the 5 founding members of CEPI. Subsequent to that, CEPI was formally announced at the World Economic Forum and has attracted a fair amount of funding from a number of governments as well as the philanthropic organizations that I mentioned. India set up a parallel program called INCEPI at the Department of Biotechnology, which was divided between the Biotechnology Industry Research Assistance Council and the Translational Health Science and Technology Institute. They had parallel goals, which was really to develop vaccines for emerging infectious diseases. Because CEPI had identified targets that it would, specific targets, it had started work on a MERS coronavirus vaccine, or rather it had funded the University of Oxford to start work on a MERS coronavirus vaccine, as well as on the use of platform technologies that would allow for very rapid vaccine development. So when SARS-CoV-2 hit, it was possible to transition many of the MERS-CoV programs as well as the platform technology programs to develop rapidly vaccines for SARS-CoV-2. So the names that you hear, Moderna, Pfizer, BioNTech, the University of Oxford, many other partners around the world were funded by CEPI in varying amounts well before SARS-CoV-2, and many have been funded subsequently as well. So I'll stop there and hand off to Andy, I guess.
Thank you, Gagandeep. Let me move on to the third speaker. But before I introduce him, let me tell all three speakers that we will go beyond 9:30. Professor Pollard may be time constrained, so it's up to him. And by popular request, we'll go from 9:30 to 10 or answering various questions that the webinar participants may have. We didn't want to delay or disturb the three speakers' questions because, time constraints. So let me introduce Professor Pollard, who chaired the famous NICE meningitis guidelines development group in the UK. He, as Gagandeep mentioned, started his work on MERS vaccine issues, and from there he quickly moved into the vaccine development for the COVID-19. He has extensive tropical experience with typhoid fever in Nepal, Bangladesh, and Malawi. And he chairs the UK Department of Health and Social Care's Joint Committee on Vaccination and Immunization, member of WHO's SAGE, that is Strategic Advisory Group of Experts on Immunization, and chaired the European Medicines Agency Scientific Advisory Group on vaccines during 2012 to 2020. Over to you, Dr. Pollard. I have a little problem with my audio, but don't worry, I will.
Thank you very much, and thanks for the kind introduction, and also to Cherry for setting the scene. I think if we cast our minds back to a year ago, which I did recently, to just think what we were all doing then, it seems like a completely different world that we were in. And I looked at an article that was in the New York Times in the first week in January, and it described cases of pneumonia in China. But it said the good news is that this virus doesn't spread from human to human. And so 1 year ago, we were in a very different position where we had no idea what was going to happen to humanity over the course of the last 12 months. And of course, for us here in Oxford, as you mentioned, there was work already going on MERS coronavirus vaccines, which meant that we really had a good understanding about how to make a coronavirus vaccine using our platform, and also the 20 years of work that's gone on before on the biology of coronaviruses related to both the SARS and the MERS outbreak, meant that all developers were in a very strong position as soon as we had the genome sequence in the middle of January for the virus to accelerate very rapidly and to design the first vaccines. And here in Oxford, we got together on the 11th of February, realizing the huge mountain that we had to climb in order to be able to put together a program to face a pandemic, and brought together the many different departments here in Oxford that work on vaccine development already, to join forces. And in that consortium here, my role was to lead the clinical development program. And so we worked over the first couple of months of the year in preclinical studies and manufacturing, and started vaccinating on the 23rd of April with the Oxford vaccine. And what the vaccine is, it's a chimpanzee adenovirus, which has been rendered replication incompetent, so it cannot make new viruses. It doesn't cause any ongoing infection in humans, so it's perfectly safe. But into that virus has been inserted the gene for spike protein, so that after immunization, the spike protein is expressed in the cells after the vaccine is given, and that induces an immune response to spike protein. And so during the course of the last year, we expanded our trials to 19 sites in the UK. We were very keen in our development program to have good geographic and ethnic representation. So I initiated trials in Latin America and Africa, and also started planning for trials in South Asia. But in the meantime, we formed a partnership with AstraZeneca, who were absolutely critical because they shared our vision for a not-for-profit distribution of the vaccine, and so made it very easy for the university to partner with them. And then, as you know, they then made an arrangement with Serum Institute of India to manufacture and distribute the vaccine in India and to many other countries. And so the trials of the vaccine in South Asia have actually been driven by Serum Institute rather than by trials directly from us here in Oxford. So that first bit of the development program was all about this vision of a vaccine which was developed globally with global partners, and to try to have equity in distribution and availability of the vaccine. And I think that's been a really important part of our goals as we've gone along during the course of the year in development. The phase 1 trials showed that 2 doses of the vaccine gave better immune responses than 1. And so our program of work switched to a 2-dose schedule. And because of some manufacturing delays, we ended up with various different dosing intervals from 1 month to about 3 months, and showed that after the second dose, particularly if the interval was longer, we saw better immune responses and higher efficacy. And so in the program moving forwards here in the UK, now that the vaccine is authorized, the vaccine is given as a 2-dose schedule with the doses given 3 months apart. So the development program in those 3 countries went very rapidly last year. And as waves of the pandemic hit the various different countries, we were able to capture the cases, and by the end of November had enough cases in the trial to be able to measure the efficacy of the vaccine, and showed that after the first dose that there's about 70% efficacy overall, and then after the second dose there's, if it's given 3 months apart, there's around about 80% efficacy. So with that program that we have in place, we were able to gain authorization here in the UK at the end of December, 30th of December. And since then, many other countries have authorized the vaccine, including India and Argentina and Brazil, several countries in various other parts of the world, Morocco, the Dominican Republic, Mexico, many countries. And we hope to have a decision next week from the European Medicines Agency. So a lot of progress over the course of a relatively short period of time. And I think for an academic institution to be able to pull together a whole end-to-end development program, which in my experience usually takes 5 to 10 years, but we've been able to deliver it in about 10 months, really, I think, gives huge credit to the team of people here in Oxford, but also the academic partners around the UK, in the sites in Brazil and South Africa, and obviously our collaborators at Serum Institute of India and AstraZeneca. And of course, a really important component of this is the willingness of volunteers to come forward, particularly early on when we have no idea about safety or about the efficacy of the vaccine. And yet tens of thousands of people are prepared to come forwards and take part in the trials to answer these important questions and help perhaps be part of a program that may be important for humanity and for controlling the pandemic over the coming year. So I'm going to actually stop there because I'm sure there'll be lots of questions. But I thought I'd just give you a very short overview of the year that we've just had.
Thank you, Professor Pollard. Can I ask you one clarification? Vasanta, in her first talk, mentioned that the Covishield and the AstraZeneca parent vaccine are undergoing a non-inferiority comparison trial in India, which immediately begs the question as to if they are not identical, what are the biological differences, if any, or is it a difference in the manufacturing process or excipients or what have you?
That's a very good question. The manufacturing process is extremely similar between the process in India and the process that's being used in the rest of the world. It's the same technology transfer for the process. So I don't think there's any real differences in manufacturing. I'm not a manufacturing expert, but casting my eye over it, it doesn't look like there's any difference. So I wouldn't anticipate anything between the two vaccines. As far as the immunogenicity studies are concerned, we've done some studies here in Oxford comparing the Covishield version and the AstraZeneca version, and they give exactly the same immune responses. And I know Serum Institute have been doing the same work in India. I think this is more of a regulatory reassurance rather than any real concern. It's the same dose, it's the same manufacturing process, so we wouldn't anticipate any differences.
Thank you. Naveen, are you there?
Yes, sir. Yes, I'm here.
Yes, Naveen is the Child Health Foundation's soul and spirit and secretary.
Sir.
Naveen, have you collected some questions from the chat box?
Sir, I think because my internet was down. Vipin, have you seen that and are you ready? I requested him to ask questions. Vipin?
Yeah. Andrew. Yeah, you see, there are a lot of queries regarding the exact duration between the first dose of second dose. You just told that in the UK trial, there was a better immune responses if the second dose is delayed. I think it was presumed to be a single dose vaccine initially when you started it. So what do you say to this, whether it should be because government of India is saying that 4 weeks between the two doses. But what your trial data has gone through, they say that after 8 weeks is a better response. What is your take on this?
Well, the trial or the license here in the UK is for the vaccine to be used at any point between 4 weeks and 12 weeks. But what we see in the trial data is that if you have a longer interval, the immune response is a bit higher and the efficacy is a bit higher. And of course, that's not surprising. We see that with many vaccines, that if you have an extended dose interval, there's more time for the immune response to mature and you get a better response to the second dose. But I think there are practical differences in approaches taken. So logistically, it can be easier to give two doses closer together in planning campaigns of mass vaccination. So I think if you were to look purely on the immunological perspective, then it may be better to have a longer gap between the doses and have a 3-month gap as we're doing here in the UK. But I think I would follow local guidance as to the right way. The authorization from the MHRA is anything from 4 weeks to 12 weeks.
Thank you. Can I follow that up with one question? Could you give a third dose later on and expect high levels of immune response, both T cell and antibody?
Well, we haven't tested that out specifically with this vaccine so far. The concern that people have raised is whether immunity to the viral vector, the adenovirus, might reduce the response to a later dose. And so one of the things that we have done in the studies is included a group who'd previously had this viral vector with other vaccines to see whether that has any impact. And so far, we've only started analysing the data, but there's no evidence that previous vaccination affects the immune response. But I think that's something that needs looking into further.
Thank you. Vipin?
Yeah. If we go your Lancet trial, you said that there was, I think it was because of certain manufacturing places that you come up with the low dose and standard dose schedule, and it fared well. Your thought on it, what was the reason behind it? Why the low dose and standard dose worked in that different way?
Yeah, I think now that we've got all of the laboratory data back, it looks like this is mostly driven by the dosing interval. So the group that had the low dose followed by the standard dose had a longer dosing interval, just because of the delay in manufacturing for the second dose. And so we think that it's not the fact that it's a low dose that's the key driver of the response, it's the fact that it's a 3-month dosing interval. And we see the same phenomenon with the standard dose as well. If you have a longer dosing interval, you see a better immune response.
Yeah. Another query which is asked that in the Lancet study, you did mention about the immune responses or efficacy of your vaccine in elderly age group. It is confined up to 55 age group. And I think you have enrolled about 70-year people also, and in due course of time it will come. What are your take on, because a lot of queries are coming whether elderly people should have it or not? And
well, when we look at the immune response in our phase 2 study, it's absolutely identical in the over 70s as well as the under 55s. So I think if you take a phase 2 population, which is the healthy elderly, it's absolutely identical. So there's no question in that group. So in the phase 3 study, we did include frail elderly individuals, and we see slightly lower immune responses in the frail elderly, but not very different. So I think at this moment, to answer the efficacy question, we need a little bit more time, because here in Europe and also in Brazil, the elderly are a bit more cautious, and so they haven't had as much disease. And so at the moment, we haven't had enough power to give absolute certainty on the vaccine efficacy in that age group. But I think that will come because the trial continues, and so we may be able to answer that in the weeks ahead. But certainly from an immune response perspective, it looks fine.
so shall the older population should wait or go ahead with this vaccine?
Yeah, so here in the UK, there's limited information, but the regulatory decision was that there's no worrying signs about immune responses in the elderly, and it's appropriate to do that. So in fact, the vaccine is being very widely rolled out in older age groups, partly also because distribution here for our vaccine is quite easy. It's stored at fridge temperatures, and the alternative at the moment that we have is the Pfizer vaccine, which is much harder to distribute because of the storage requirements for that vaccine.
Okay. Dr. Jacob John?
Go ahead, Vipin.
Okay.
More questions?
Yeah, there are a lot of queries regarding the new UK variant and this the impact of vaccines, whether these mutants have evaded the vaccine which is in use. What do you think of this Oxford vaccines? Whether it will withstand this pressure induced by the B117 in UK or in South Africa strain which is there?
Well, the truthful answer at this moment is that we don't know. But if you look at the structure of the UK variant, then it's likely that even if it has a bit of an effect on the immune response and some evasion, it's unlikely to completely evade immune responses. So we would anticipate that we still have efficacy against the UK variant. I think there's much less certainty about other variants from other countries where there are a lot more changes in spike protein, and there's some recent data in patients in South Africa, so convalescent sera, suggesting that the South African variant is able to evade the immune responses made after infection. And so I think we really need to have some good data for the vaccines to see whether vaccine immunity is also evaded. I think a really important point here, though, is that what most of the clinical trials do is measure efficacy against relatively mild infection. But what we really care about as physicians is whether the vaccines stop patients going into hospital and dying. And so one thing that we may be in a position is that, as with other coronaviruses that we see in children normally, through adult life, we still have mild upper respiratory infections with them, but we don't have severe disease. So one of the key questions for me is whether the vaccines are able to induce good T-cell responses and binding antibody responses that protect against severe disease, but perhaps because of a bit of evasion of immunity, which the viruses do, we still see mild disease or asymptomatic transmission. And those are very difficult questions to answer in the trials, because to have hospitalization at the endpoint, you need a much larger trial. So we'll probably only learn that during the rollout of vaccines in countries that have these new variants.
Jacob, there's a lot of confusion regarding a viral vector vaccine, and I'm going through the chat box. Some person has asked whether it is a DNA vaccine or it's a viral vector vaccine. What are the differentiating features between the two? Because this vaccine is also traversing through the nucleus.
Well, in some ways they are DNA vaccines, but in the end, all of these vaccines are protein vaccines. What they're all doing is leading to the expression of protein, and that then allows an immune response to be made. So the RNA vaccines, their endpoint is protein. The viral vectors, the endpoint is protein. And the DNA vaccines, the endpoint is protein. So really, as far as our immune system is concerned, there's a similarity between all of these vaccines. The difference is just the way in which the genetic material is introduced into the cell to drive the production of the protein, the spike protein, and the danger signals that brings to the immune system to get it to switch on. But in the end, it's the same immune response that we're seeing across the different platforms.
So
let me follow up that with one question, that when the cell expresses the spike protein, is it displayed on the cell surface, or is it shed into the extracellular space? Do we know that?
Yeah, it's expressed on the cell surface. So if you look at the cells, you can see the expression of protein.
Thank you. That's why it's stimulating T cell response, Th1 response, right?
Exactly, yes.
So Vipin, can I ask a question, because that's on very popular demand from all the seniors, that because one of the manufacturer has issued guideline that those who are on blood thinners, or taking aspirin or anything, should not take vaccine. It's contraindicated. So everyone is calling and asking, and can you just specify specific contraindications, and those who are on medications or immunocompromised or have other diseases, cancer survivors. So many questions are related to allergy, and even like there is one advisory from other company that anybody who has allergy should not take vaccine. So what about maybe at least you can answer for Covishield?
Yes, so as far as safety is concerned in the context of various blood thinners and people with bleeding disorders, I think that's no different from any other injection. So if someone has a significant bleeding risk, then intramuscular injections are relatively contraindicated, and the recommendations that are usually used is to give those vaccines subcutaneously. But in this context, we don't have any data on subcutaneous administration. So I think that's why there's been some caution in that environment. But for many of those diseases, people who are on blood thinners, the bleeding risk is extremely small. And so you have to have a discussion with the individual clinician about what the risk is for that patient and whether the risk benefit of giving an intramuscular injection is in favor of giving the injection. So I don't think it's an absolute contraindication, and you can't really generalize across all different patient groups. As far as allergies are concerned, the particular issue that has been raised over the last few months is with the Pfizer RNA vaccine, which contains polyethylene glycol. And it's known that some people will have allergies to polyethylene glycol or PEG. In the Covishield vaccine, there is no PEG, but there is a related compound called polysorbate 80. But that's found in many different vaccines, including some of the flu vaccines that are used here in Europe. And we don't see an excess rate of anaphylaxis with it. So we wouldn't expect that. And certainly there haven't been reports here in the media in the UK, despite large numbers of doses, millions of doses being rolled out. So I don't think there's a particular allergy risk. But of course, as with all vaccines, if someone is allergic to a component of the vaccine, then they shouldn't have it. If they get anaphylaxis, I should say. But there's no absolute contraindication in people with other types of allergies.
Let me add to that, Vipin or Naveen, that anybody on anticoagulant treatment or even clopidogrel, the platelet aggregation inhibitors, if they have something to do with tooth work, they go off the treatment for a few days, maybe maximum of 5 days, because many of them have long half-life. And then after the dental work, 2 days later, you restart your blood thinning therapy. Similarly, if you're afraid of an intramuscular injection producing severe bleeding, that's exactly what you should follow. Take a break from the, many of them are taking because of atrial fibrillation to prevent clotting or myocardial coronary artery. So a short break will not do any harm, we hope. So that's the recommendation for tooth work, dental work, and the same thing can apply for intramuscular injections, and of course, apply pressure in the common sense approach. So other than that, there is no absolute contraindication for these drugs that are being used.
Yeah. Andy, one question regarding this manufacturing process of this Oxford vaccine. You see the Johnson & Johnson is also developing one vector vaccine, this adenovirus 26. Do you think they have adopted a different stabilization process of spike protein to maintain its pre-fusion conformation? And they have used these 2p mutations. And similarly, the Moderna people as well as the Pfizer people, and I think Novavax also, they have utilized this technology to maintain, to stabilize, because protein is a rigid amino acid. This stabilize the spike protein in proper conformation. So it is expected that it will have a better immune response if it is upright, this RBD is upright. Do you think it has something to do with and what is your take on your development of your vaccine, whether you looked into this aspect, and does it have something to do with the immune responses?
Well, I think it's a good question. Of course, we don't know the answer to that. There are no head-to-head studies at this stage to address the question. And although I think Johnson & Johnson will be presenting their data very soon, we haven't seen it yet, so we can't really make any judgment on it. They've got a different viral vector, Ad26, for their vaccine, which is in many ways is similar to the chimpanzee adenovirus we're using. But I don't know what their responses look like yet. I would imagine that it should be fairly similar, but I think we need to see the data before speculating about it. And I think this question about whether different forms of the protein and whether they're stabilized in pre-fusion or whether they're natural is an important scientific question which we need to address. And of course, one of the things is that with the full-length spike protein, some of it will be in pre-fusion form anyway, and some will be in post-fusion even without stabilization. But I think it's a good question, but we just don't know the answer to it.
Thank you. I think there's a lot of questions regarding this one complication, neurological complication, which occurred in Oxford trial, in UK trial, which is I think transverse myelitis. And I think there are 2-3 cases. One is come across in India also, but it was not probably related to this vaccine. A lot of apprehension regarding this neurological complications associated with the Oxford vaccines. Do you have anything on this to add?
Yeah, I mean, so there's only one case of transverse myelitis where the independent neurological review said that they couldn't tell whether it was related or not. And of course, when you have events that occur in a phase 3 trial that don't have an obvious cause, you can't be sure whether or not they're related to the vaccine. And so you have to flag them as potentially important and to monitor thereafter. But we haven't seen any further episodes that have occurred in the US trial, for example, which is another 30,000 people. And so far, there's been no reports in the rollout of the vaccine. But I think that the whole purpose of collecting information like this in clinical trials is to make sure there's very careful monitoring of safety. And if there is a signal, if you have multiple events in the vaccinated arm, and there's a signal of something you should be looking out for and worrying about. But having a single case doesn't really help you. It says there's something we should be thinking about, but we haven't proven causality at this stage. And, for example, with the Pfizer vaccine, they had 4 cases of Bell's palsy in the vaccine group, but that doesn't prove for certain, even though that's 4 versus 0, it doesn't prove for certain that that's an association of the vaccine. But it is a flag to say that we need to look for these types of neurological complications in case there is a signal when you have larger numbers of people vaccinated. And that's really the approach the regulators have taken, that they don't see any clear signal, but if there's a case and it's unexplained, you have to take note of that and follow as you roll out the vaccine to see whether there's some other safety concern. But I don't think anyone's concerned at this moment.
Thank you.
Vipin, let me add, Andrew, I have a case record with me of another neurological problem with the either Covishield or the AstraZeneca vaccine, because they were doing vaccine comparison. And I will correspond with you privately. I will let you know the details so that you are aware of the details of it. It's also an unexplained neurological illness, completely recovered, but no etiological association with anything, not bacterial, not viral. And there is a suggestion of a temporary autoimmune kind of a disease, neurological disease. I will.
Yeah. I think, of course, in clinical trials, these things, stuff happens to people. So we do see in when you have a large trial with tens of thousands of people, there will be odd events that happen. And the difficulty is discerning whether or not they're related to the product. And I think that's something we can only do well by having ongoing good quality surveillance, and particularly once you roll out a vaccine in population. So I don't think these are so far signals that tell us there's a problem, but they are telling us that we, as always, we need good surveillance programs for safety monitoring.
So, Dr. Andrew, are there any unexplained or or any deaths which are which can be associated with causal relations with vaccine during trial or after that, or which are unexplained?
Well, there's no vaccine-related deaths in the clinical trials. That I know because I've seen all the data. As far as rollout is concerned, I don't know the data. That's monitored by Public Health England here in England. So because it's only just a few weeks since rollout started, I haven't seen any data.
Do you have any comment on the Swedish experience of very old people whom they call fragile or frail with the mortality pretty soon after the vaccine? That's Pfizer vaccine. That's mRNA vaccine.
Yeah, so there were some reports from Norway about what they considered to be an increased number of deaths in some of the very frail elderly in the few days after the Pfizer vaccine. But from at this moment, I don't know whether that is that those were extremely elderly, frail individuals who may have died anyway, or whether there's an association. So I think we'd have to ask the Norwegian Public Health Agency for more detail on that.
Vipin, Naveen?
Maybe I can comment on the Norwegian data because that was reviewed by the subcommittee that has been set up for safety of COVID-19 vaccines under the Global Advisory Committee for Vaccine Safety, and I'm a member of that. We've just put out a statement saying that at the moment, we don't think there is any signal that is of concern, but this is something that we will need to continue to monitor.
Would you say that very old people, if they are frail and sick, and they should better be cocooned and infection prevented, everyone else in the vicinity vaccinated, and they be protected through such interventions rather than rush with the vaccine into them?
I think that the difficulty is that the evidence that we have so far is that it's extremely difficult to protect the most vulnerable in society. And I think the only way we can do that is through vaccination. We don't have very convincing evidence that cocooning as a strategy will work. The frailest and most vulnerable in society need to interact with other people to look after them. And those individuals, even if they're vaccinated, it's possible they can still transmit. There won't be 100% protection. So I think we have to vaccinate the most vulnerable as our absolute priority.
Andrew, there are questions regarding related to reinfection. And what is your take on the duration, probable duration of protection of these vaccines, particularly the new generation vaccines, genetic vaccines? You see, most of trials have gone in UK, in the healthcare professionals. And I was going through these silent trials, and many other trials, they have shown that up to 6 months, the chances of protection with the natural disease is much more. And they've shown around 83% efficacy of natural infection. What is your take on this reinfection issue and duration of protection of these vaccines?
Yeah, so I mean, it's a really interesting study, that one you mentioned, which shows that for 5 months after natural infection, there's about 83% protection. So I think that tells you if you get a strong immune stimulus from infection, that's the sort of level of protection you can expect. So with vaccines, of course, there may be a different trajectory of the immune response. We tend, after viral vectors, to see quite stable levels of antibody, at least during the first year. But I think we, from a scientific perspective, we ought to hold judgment until we've got the data, rather than making bold claims at this stage. I mean, we just don't have enough time.
You see, there are a lot of queries regarding which is better, the natural infection, antibodies induced by natural infection and by the vaccines. You see, I've gone through these trials of new generation vaccines, and their induced antibodies production and vis-a-vis to the convalescent sera. So they are not to the tune of 2 folds or 3 folds or 5 folds. They're slightly higher in some candidates. What is your take on this? Whether the natural immunity is going to be better than the vaccine induced?
I don't know the answer to the question. There are different antibody functions after vaccination than there are after infection. We put those in a Nature Medicine paper last year, which describes the spectrum of different antibody functions. And it's very different after 2 doses of the vaccine when compared with after natural infection. So I think all that tells us is they're different, but it doesn't tell us whether that's better or worse. So I think we need time to understand whether having those different functions is going to give better persistence or a difference in the quality of protection over time. So I think we just have to hold judgment at the moment.
Thank you. One more question, which is there in the chat box. A lot of people are worried that they have already suffered a COVID infection. At least 3 months, 4 months, and they are still having anti-spike antibodies in their serum. So, and many of them, they have received immunosuppressive drugs like steroids or IL-6 inhibitors. So what is your advise to them whether they should go for vaccination and how long should they wait? It should be immediate or some time duration should be kept between these two.
Well, in all of the trials, the regulators asked for inclusion of people who'd had previous infection based on serology. So we do have quite good evidence that vaccines, all of those being tested that I'm aware of, that they are safe in individuals who've got previous infection. But mostly what was included in the trials are people with mild infection or asymptomatic infection. And we see in those situations that people often will get a boost in their antibody response as a result of vaccination. So I think that's the first bit of evidence. There's a suggestion in the data that people who have had previous infection may get slightly more reactogenicity when they're vaccinated. So they might have more of a fever or sore arm or a headache when, on the day after they've been vaccinated. But otherwise, they seem to be perfectly safe. I think there's a slightly separate question which you're asking is what about the people who've had more severe disease? And that experience is currently being gained at the moment in the UK. And I'm not aware of any safety signals, but there are some anecdotal reports of people, again, having slightly more sore arms or headaches or feeling feverish after they've been vaccinated. And currently the recommendation is to wait at least 4 weeks after infection before being vaccinated.
So, Dr. Andrew, let me take this opportunity to ask, which is concerning me. I took the Covishield on 16th and there was no side effect. So what's the problem? Vaccine is not working or it is known?
Well, in the trials, there's certainly some people who have very mild side effects. I was vaccinated 3 weeks ago and I had mild side effects. I just had a bit of fatigue for a couple of days. And so it's very common, particularly if you're a slightly older adult, to have very mild symptoms. It seems that with younger adults, and I think it's true with other vaccines as well, that the reactogenicity is much greater. So I think the best thing is to, with Covishield, is to be over 55 if possible, and the reactions are a bit lower. And also with the second dose, the reactions are a bit lower. So I don't think I'd be worried about having no reactions. I think you've been very lucky.
Okay.
Jacob Johnson.
And I should just add that even in people without any reactions, we've seen good immune responses in the trial. We haven't seen a relationship between the two so far.
Dr. Andrew, there's a lot of apprehension regarding this nucleic acid-based, particularly the DNA and viral vector vaccine, that there are remote possibility of integration of genetic material with the DNA, the sort of with the genome, since it travels through the nucleus. Any chances of that happening, and for how long one should wait for this? Because most of the side effects are expected within 1 or 2 months, which is the usual trial period. Yeah.
Yeah, so that's right. So almost all side effects happen within the first few days. I mean, this viral vector doesn't integrate into the human genome. The virus itself doesn't replicate. It doesn't make any more copies of itself. It doesn't have the machinery to integrate into the genome. And lots of work has been done over the last 20 years to try and address that question specifically. And there's no evidence that happens. So I think the regulators look at this type of stuff in great detail, and there's not been any concerns about that.
Thank you.
Vipin, may I ask a question?
Yeah, yes, sir.
The chimpanzee adeno, how many cycles does it replicate in the human being? 1 cycle or a few cycles or none at all?
None at all. So all that happens after immunization is that the genes are expressed to get spike protein expressed, and that's it.
You see, many replicating viral vector vaccines are in the offing and they are in the various stages of development. How do you see whether they are going to have a better efficacy and immune response in comparison to a non-replicating viral vector?
I think it's difficult to know because each viral vector is different in its replication competence of the replicating ones, and also the dose of them that's used. So I think until we've seen some data, it's hard to answer that. The VSV Ebola vaccine that's made by Merck has been widely used in Africa, has been shown to be very safe and to induce good immune responses. So I don't think I would be concerned about the replicating ones, but we do need to see the data from the trials.
Okay, thank you. Dr. Khan, would you like to ask something?
No, I have no more questions. Anybody? What time is it now? 9:50 here. It's time to wind up. Naveen?
Yeah, I don't know. Dr. Kang has not finished. Maybe she wants to speak. Dr. Gagandeep, you want to say anything?
No, I have nothing more to say. If you have questions for me, I'd be happy to take them.
There are many questions, but I think you can see the chat box, and if you feel like answering anything, there are many questions.
That's not fair to the panelists. Let's wind up. Naveen, would you?
Yeah, Dr. John, I just, for one question, I would like to just an answer. Many people are asking questions that can we take 2 different vaccines? Will take first dose COVID and then second vaccine Covaxin. Can we do that? Number of people are asking this question. I think it's said very clearly in the fact sheet of both the vaccines, and then you can't mix up vaccine. If you've taken one vaccine, the second dose should be the same vaccine, because unless the second dose is taken, the vaccine is not going to produce effective immunogenic reaction in the body. So if you just say it, because these are different vaccines, one is a chimpanzee adenovirus, the other one is the inactivated coronavirus vaccine, and if you are going to have 2 different vaccines, and then you are confusing the system, finally both will become ineffective. So I don't know, number of people have asked this question that can we have both the vaccines? You can't have both the vaccines. Whichever you have taken first dose, please take only that second dose of the same vaccine.
It's a really important question, and I think there's 2 aspects to this. One is the biology, and this is very clear that the main immune response is against spike protein. Both of these vaccines contain spike protein, so biologically you would expect that if you've had one of the vaccines, the other vaccine should boost it, but we don't have the data. And normally in making policy and from a regulatory perspective, you would try to generate the data before switching to a policy and where you recommended that happened. So I think although we think from a scientific point of view, it should be okay, it would be nice to actually have some data to show that. And I think you're in India, you're in a perfect situation with both vaccines available to rapidly address that question. I think the other bit, when you sort of stand back and think about what does the physician do in the clinic, if you get a supply 1 month of Covishield and the next month it's Covaxin, should you deny your patients their second dose or take a pragmatic decision to give the dose that's available? And here in the UK, the decision has been in normal circumstances, you should give 2 doses of the same vaccine. But if you're in a supply constrained situation where you're in a clinic, there's nothing else, rather than denying that person their second dose, use the second dose of the vaccine that's available. So that's the UK policy decision, which is a pragmatic one. Try to stick with the same dose, but don't deny people the second dose if they're there in front of you.
So one more question. Suppose we are taking Covishield now or Covaxin now, and future mRNA vaccine becomes available in private market. Can we take, even if we have taken 2 doses of Covishield or Covaxin, because that's the plan of, many of in India, that, let us settle for the 70%, and then once we get 90%, why not to get that? What about that?
Well, I think really when we think about the sort of global equity question, what you need is for the really constrained number of doses that we have in the world, everyone should have a chance of getting one of those doses. So I think if people are doubling up, then they are potentially denying others getting their first dose or their second dose of the vaccine. So I would find it hard to support that personally, because we've got 7 billion people on the planet who need protecting.
Not now, but in future, suppose some other vaccine become available. What about the safety? And the main thing is safety. Can you take it or not?
I don't really see that there should be any safety concern in the future if that were to happen.
And what about the one dose, any protection after one dose of Oxford vaccine? Any data to support that?
So the regulatory analysis that was done by the MHRA indicates that between the first and the second dose, there's about somewhere around 70% protection up to 3 months.
Yeah, Andrew, there's a question regarding this priming with one type of a vector and boosting with another booster. I think the Oxford vaccine people, they are conducting one trial in Ukraine with the idea of priming with this Oxford vaccine and boosting with the Sputnik adenovirus 26.
Yeah, so that's a study being run by AstraZeneca, as you say, to prime with one of the vaccines, and I can't remember which way around it is now, but with the chimpanzee adeno, and then boosting with the Ad26 vaccine from the Russian Institute. Here in the UK, we're doing studies mixing and matching with our vaccine and then the RNA vaccines to try and see what if you give a first dose of chimpanzee adeno and a second dose of the Pfizer vaccine.
Thank you. There is another question regarding this efficacy of inactivated vaccines against this mutant strain, for example, the UK strain. And there are a lot of questions during the various scientific sessions, and the producers of this Covaxin, they have claimed that our vaccine is going to protect against the drifted UK strain also. Your comment on this?
Well, at this moment, no one has demonstrated any data around the UK strain. We're certainly hoping that from our trial we'll be able to do that soon. We're just waiting for enough cases to accumulate, and obviously we have to sequence the virus in each case and to be sure that which variant it is, whether it's the new variant or the old variant. But I think at the moment we can't really address the question of whether the vaccines provide full protection. I have to say that we really expect that against the UK variant, there shouldn't be a big problem because the changes in spike protein are fairly minor.
Okay,
but I think in this question, Dr. Vipin, I think actually, I think in one of the interviews, I think Dr. Krishna Ella has said that this is only a hypothesis, that since it is an inactivated whole virus, probably since the mutation is not a very severe one, probably it should be, it's only, he himself has said that it's only a hypothesis. I don't think they're actually claiming, but this is a, people think that probably it will work.
They are not claiming, but on their behalf, many experts
claiming,
I think we need to wind up because we are, Dr. Pollard has given only half an hour, but it's still, so what we promised, we will put this on YouTube as someone has requested. We will try to get answers for all your questions and maybe see how we can reach to you and, but just go on looking for on Child Health Foundation website and YouTube and Twitter handle, and we will try to answer all the question and put this on definitely this whole recorded version on YouTube. So thank you so much. Over to Dr. Jacob John.
Naveen, over to you. You can say a word of thanks to each speaker, panelist, and then we'll wind up.
Definitely, I joined late, but thank you very much, Dr. Vasanta, Dr. Gagandeep Kang, Dr. Andrew Pollard, and all the, such a wonderful response to our webinars. And we promise you to continue same in as we go along. And thanks, Dr. Jacob John, to you also, because, for this is planning this so well. And I think everyone has enjoyed our last webinar also, this webinar also, and we will continue. Thank you so much. Continue to vaccinate. Most of us are already vaccinated. And and just post, make a video or post on social media, so we can, increase the vaccine confidence. Thank you so much. Thank you and good night.